Good morning, everybody. We're going to get started. So I have the pleasure today of welcoming Dr. Chuck Murry. So Dr. Murry is originally from the Midwest. He did his undergraduate degree and graduated Phi Beta Kappa from the University of North Dakota. He then went on to Duke University, where he completed his MD PhD and wrote his thesis in mechanisms of cell injury during myocardial ischemia and reperfusion. He then went on and did his residency training here at the University of Washington, where he then stayed on as faculty and currently holds multiple appointments as professor in the Department of Pathology, Bioengineering, and Medicine and Cardiology. He is also the director for the Center for Cardiovascular Biology and the co-director for the Institute for Stem Cell and Regenerative Medicine. He does an enormous amount of research. He has tremendous grant support, over 130 peer-reviewed journal articles, a dozen book chapters, and has been an invited speaker to nearly 200 seminars. He also has recognized locally, nationally, and internationally for his work. He's received numerous awards and honors. He's involved in many, many committees, both locally and nationally. And back home, he's also very dedicated to his students and trainees. He teaches many courses here at the university and has mentored dozens of trainees over the years. So I'd like to welcome Dr. Chuck Murry today for his talk on stem cells, tools for human genetics, and heart regeneration.
Good morning, everybody. Thank you for that nice introduction, Rachel. It was a little overly generous, but I'll take it. It's fun to address the Department of Medicine here in the august setting of Grand Rounds. I'm getting old enough now that I see several of my own physicians out in the audience, so it's kind of fun. But it's as a pathologist, you know, we know where this is all heading. I guess as well. Anyway, glad to be here. And what I'd like to do this morning is to share some of the things that have been going on in my laboratory, and then finish with a little story about what's going to be going on in terms of, we hope, translating some of these findings towards regeneration of human hearts. Disclosure, we've founded a biotechnology company here it's a University of Washington spin-out. It's called Beat Biotherapeutics. Here's just a quick schematic of what's going on in my laboratory. Right now, we've gotten really very obsessed with the biology of pluripotent stem cells. And pluripotent cells are the ones that can turn into virtually everything in the body. And so this would be the embryonic stem cell, or the reprogrammed equivalent, the induced pluripotent stem cell. We have gotten quite good over the years at turning them into cardiac muscle cells. And a fraction of the laboratory studies what the biology is of differentiation. So we're interested in sort of the nuclear biology. How does this happen? How does the DNA architecture rearrange itself? Patterns of gene expression change? What are the regulatory molecules, both within the nucleus and within the, from membrane to cytoplasm, that are involved in that? And principally, this is driven by sort of omics level, big data sets, that kind of stuff. And the idea is to really understand how cardiac muscle is programmed in the first instance, and then maybe gain some insights into how we may reprogram other cell types into cardiac muscle. Once we have cardiac muscle, we are interested in doing a couple of things with them. We use them as therapeutics, both as in cell transplants and in tissue engineering. And a chunk of what I speak about today will be using cells as therapeutics. And the other part, the most recent area that we've gotten into, is using induced pluripotent stem cells, or reprogrammed stem cells, to get insights into genetically based heart muscle diseases. And we're going to start with cardiomyopathy. And with the idea here is to use stem cells to really understand genotype-phenotype correlations in a much better way. Down the road, to use technologies of genomic editing for gene correction and drug screens and that sort of thing. So let's just jump in. So it's going to be these two right-hand sides of things that I'm going to be speaking about today. The biology of differentiation would have to wait to another time. So this is my cold to Newcastle slide. Heart failure is a burgeoning epidemic. Everybody knows this. It's bad. You don't want it. We've got about 6 million Americans walking around with heart failure right now. They're not walking too fast, but they're hanging in there. I think people are also familiar with the statistic. It's grim. Once you get a diagnosis of heart failure, considering all comers without stratification, you've got on average about 5 years to live. And contrast that with something like breast cancer, which is another bad diagnosis that you don't want. But on average, you've got about 11 years to live. So it's a serious illness. It's a disease of aging, of course, and our population is aging as my generation of baby boomers ages out. The young folks are going to be supporting us as we go along. Thank you very much for that. And it's already the #1 cause of hospital admissions in Medicare patients. And it's the #1 cause of medical expenses basically in the United States and all developed world. And the root cause of heart failure, irrespective of how one gets there, is that there's an insufficiency in muscle cells. So like many chronic diseases that we face in society, the root cause is deficiency of a key population of cells. So the hope is that somehow we can harness stem cells in a way that we can repopulate the heart. In this instance, other people are interested in repopulating beta cells for diabetes treatment or retinal cells for blindness and that sort of thing. But our passion is to figure out what's wrong with the heart and how to fix it. This is, of course, familiar to everybody. I'm a pathologist, as you heard. So this is a coronary thrombosis from a patient who died at the University of Washington Medical Center. She died in the acute phase of her illness, and what you see is a ruptured atherosclerotic plaque with an occlusive thrombus in the lumen of the vessel. But cardiology has gotten quite good at treating this acute illness. And the vast majority of patients are successfully reperfused now with a combination of thrombolytic agents and mechanical interventions like angioplasty and stenting. And so the mortality for acute myocardial infarction has gone down. In the process, we've created this new problem of heart failure. And so in a way, it's kind of an iatrogenic problem because people used to just drop dead. And by the way, that was really cheap. If you're just a health economist and you're just a bottom line driven sort of person, sudden death is really economical. And it's this lingering, not quite hitting on all cylinders that's expensive. And so I'm being a little snarky, but I do get a little weary of only hearing about economic bottom lines, and actually, I suspect we do a little good by keeping people alive as well. That's an editorial comment. This slide is a heart of a patient who had an aneuryseptic myocardial infarction, also from the University of Washington. So what you see is this big fibrotic region in the anterior wall extending throughout most of the interventricular septum. There's big-time hypertrophy in the posterior wall of the heart, or the inferior, as cardiologists would call it. But the heart is essentially the least regenerative organ in the body. It really has a minimal ability to generate new cardiomyocytes after injury. And as a consequence, it heals by scar formation rather than muscle regeneration. And the consequence of that is that there is a marked decline in function after a myocardial infarction. So this is the basis for most causes of heart failure, but we also have hypertension, valvular disease, cardiomyopathy as an increasingly frequently recognized cause as well. By the way, I like to ask this of the medical students. Is this patient alive or dead? Got the heart here, right? So it's a trick question, of course, because I always try to suck the med students. And this is one of our transplant patients. And this person is alive and doing well, and we're regularly doing surveillance biopsies on his new heart. But heart transplantation, as Ron McClellan says, is a boutique industry. We do about 2,200 hearts every year in the United States, and with a population of 6 million folks with heart failure, really less than 1/10 of 1% of patients who could potentially benefit from it. So we're never going to transplant our way out of the heart failure epidemic. So what I think we can do, however, is find ways of developing stem cell approaches, because stem cells are a virtually inexhaustible, completely renewable resource that can be expanded up by the billions, and we've done so, as I'll show you in a little bit. And we've learned how to turn these embryonic stem cells, in this instance, into cardiac muscle. So this is a movie taken through a phase contrast microscope where we've got a sheet of human cardiac muscle. This is probably 70 or 80% cardiomyocytes. There's some other cells that come along for the ride in the differentiation process that are mostly fibroblastic type cells. And so that looks pretty cool. You can see these sort of spiral waves, that sort of thing. And when we get good preps, you can actually see it with your naked eye. This is a 10-centimeter Petri dish, about yay big, and you can see just using this low-tech, sit on the floor and shoot up at the ceiling, and people look over, is this thing on? But you can see waves rippling across the Petri dish like waves on a pond. So a dish like this full of human cardiac muscle would have somewhere between 20 and 50 million cardiomyocytes in it. Just to do some back-of-the-envelope kind of mathematics, if you have a myocardial infarction that's clinically significant, that would kill off about 1 billion cardiomyocytes. So somewhere in the order of 50 dishes like this would have the equivalent of what is lost in a myocardial infarction. And so you can see that, it's feasible to be talking about generating enough cardiomyocytes, even just using brute force, plain flat tissue culture approaches like this, to generate enough cells to start thinking about taking a shot on goal for a clinical trial. So I'm going to tell you two stories today. The first is one that was led in my laboratory by Daniel Yang, who's a cardiology fellow. He was a research first fellow, and he's just gone back to the clinics now. And regrettably, Daniel is off to Chicago for a young investigator competition, couldn't be here today. But what this is about the tools for human genetics and the insights into genetic diseases that I think we're going to get from stem cell biology, and we're going to use a familial cardiomyopathy as a test case. So I'd like to introduce this family that has a mutation in the gene called myosin heavy chain 7, also called beta-myosin heavy chain. This is the principal contractile protein of the heart. It makes up maybe 10% of the dry weight of the heart. It's the big myosin of actin and myosin with the sliding filaments and all that sort of thing in the human heart. The proband in this instance, the person who came to our attention, was initially the father. The family came to our attention because of the kids through being screened at Children's Hospital after dad had been identified. And so this was a new mutation in myosin 7, a glutamate to glycine mutation, so a non-conservative from a charged acidic amino acid to a neutral amino acid, so a non-conservative change. And the dad was enrolled in our study. We did the skin biopsy, and unfortunately he passed away subsequently from sudden death after that. His sister had also had marked systolic failure and needed a total artificial heart. She was never genotyped, but we think she was very likely to be affected. And then, based on genotyping, 2 of the 3 children who came to our attention at about age 18, they're about 20 years old now. They have had normal ejection fractions, but if we use more subtle approaches to look at wall motion, say regional systolic wall thickening, it's impaired in these kids. Radial or circumferential strain, rather, is also impaired based on Doppler. And so we think that they have subtle phenotypes that will become manifest later. So we reprogrammed induced pluripotent stem cells from these patients. I'll show you how we did that in just a moment. From the dad and 2 of the kids, we used mom as a control and then 2 unrelated wild-type controls as well. There were a few challenges to doing this. We didn't think too much about it. We just jumped into this, but we might have taken a different approach. But we ended up studying a new mutation, something that had never been described in the literature, rather than one that had been mined out extensively with animal models made and that sort of thing. But whatever, we did it. And this isn't a classic hypertrophic or dilated cardiomyopathy. They have impaired systolic function, but for whatever reason, their ventricles don't dilate. People in cardiology see this oftentimes, that you have impaired systolic function without dilation. And so people always want to, I'm a pathologist. I like to stick things into bins because I'm a diagnostician, I guess. But this doesn't really fit precisely into either of those camps. And the final challenge with this is the severe disease has its onset in adulthood. The cells that we can generate from stem cells are immature, so it's a question of, can we actually spot the phenotype in immature cells when it's adult onset disease? All right, so let's just talk a little bit about myosin. Myosin makes up the thick filament of the sarcomere. People, I think, are probably familiar with that. It's got a rod domain, a hinge domain, and then these 2 necks that are actually involved in binding actin. The region where a mutation resides is in what's called the S2 domain of this neck. This interacts with another protein on the thick filament that's a bit of a mystery still. You'd think we would have worked out everything in the sarcomere, but it's a protein very abundant called myosin-binding protein C. Myosin is the most common cause for cardiomyopathy mutations, at least for hypertrophics, and the second most common in the United States is myosin-binding protein C. So it interacts with this region called the C1, C2 domain of myosin-binding protein C. And a cartoon, here's your thin filament, your thick filament is down here at the bottom. The notion is myosin-binding protein C interacts in some fashion with the C1, C2 domain binding to the S2 domain of myosin heavy chain. This is a solution NMR-based structure looking at this interface. So on the top, these are ribbon diagrams showing myosin-binding protein C. On the bottom, we're seeing myosin. And there's a series of amino acids that make this charge-based interaction where the 2 proteins glom onto one another. And here sits our mutation, this E848. So the notion is maybe this is at a critical place for a protein-protein interaction. And in fact, a different cardiomyopathy had been identified on the corresponding face on myosin-binding protein C by J. Carter Ralph's group at the University of Wisconsin at the E258 amino acid of myosin-binding protein C. So it seemed like maybe all of these amino acid interactions are critical. You disrupt one, the protein-protein interaction falls apart. At least this is our hypothesis. So we expected if we made cardiomyocytes from patients in this family that they would have impaired contractile function, and if we found biochemical ways to look for protein-protein interaction, between myosin and myosin-binding protein C, that this interaction would be impaired. So we use this remarkable approach called reprogramming to generate these pluripotent stem cells from the patients. And many in the audience already know about this. I'll just say this very briefly. It was discovered by a Japanese scientist named Shinya Yamanaka. And I think it's like the coolest thing since the discovery of DNA practically in biology. What Dr. Yamanaka was able to do is to take adult cells, skin fibroblasts is what he started with, and by popping in a variety of different genes that he hypothesized would reprogram these cells, he actually got to wipe the slate clean, erase their fibroblastic identity, and took them down to a ground state that's essentially an embryonic stem cell. He did this in mice, and he did this he demonstrated the following, which I thought was quite impressive. He took tail-tip fibroblasts from a boy mouse and from a girl mouse. He reprogrammed them, made them into things that look here morphologically like a embryonic stem cell. And he took those cells and injected them into the embryos of mice, and they contributed to all tissues. So they made chimeric mice, including egg and sperm. And then he took the eggs and he bred those mice together, and they generated baby mice whose grandparents were tail-tip cells. And so, damn, that was pretty impressive, I thought. And so this seems like the real, if you could make whole mice, I mean, we could probably, Brave New World, we could probably make whole people this way, that sort of thing. I think that's a bad idea, but it's really quite potent. And so the idea is, if we take this from the standpoint of genetics, we should be able to reproduce genetic diseases in a dish, because if we start with the patient, the stem cells and then the cardiomyocytes derived therefrom should contain the full genomic complement, including the mutations in myosin in this instance. So that's what we did. And I'll skip through all the pain and suffering. We made the cells, we turned them into cardiomyocytes, and we looked at the cardiomyocytes that we got. We looked at the wild-type ones, we looked at the mutant ones, and we looked at how much they were shortening in cell culture, and we didn't see much difference. So this was a disappointment. So we thought, well, if anybody who has cultured cardiac muscle cells knows that the cytoskeleton, the contractile apparatus of these cells is totally disheveled. It looks like the worst hypertrophic cardiomyopathy you'd ever imagine. They're totally disarrayed. So we thought, well, maybe if we align the cytoskeletons of these cells, that we'll be able to have better force vectors. So we borrowed a piece of technology from a colleague in bioengineering named Deok-Ho Kim, and Deok-Ho makes what he calls these nanotopographic patterns. They're just thin ridges and grooves along a surface. So this is made out of a polymer using a technology called capillary force lithography. But just think about it as small features that are kind of on the order of a collagen fiber in terms of size. And just like the extracellular matrix guides morphogenesis for normal cell shape and that kind of thing, we think that these kinds of things will provide structural cues for the cardiomyocytes. And so if we look at a cardiomyocyte, and these are both patient-derived. This is one grown on a flat surface, just glass, and you can see, again, it's all disheveled in terms of the organization of its contractile apparatus. And here's what they look like grown on a nanopatterned surface. So you can see they have these now long cables of myofibrils, and we thought, surely this will give us better vectors for force production, and this should help bring out a difference between the mutant and the wild-type cells. So here's the difference that we found after we aligned their cytoskeletons. No, I'm not going to push another. We found no difference between them once again. So I was like, that's Daniel's getting kind of depressed. He's looking at the training grant. He's saying, I'm going to bring the training grant down if we don't get a paper out on this. So we're trying to figure out what to do next. So then this gets back to the issue of this being an adult onset disease. These are cells that are equivalent of sort of the septating heart in embryological development. They're really very early. So the idea was then perhaps if we just aged them out in culture for a little bit, we would see separation in their contractile function. And lo and behold, we did. We simply aged the cells out to between 60 and 100 days in culture. And what you're seeing, and this is a twitch, and so it gets shorter and then it relaxes. And what you can see is the wild-type cells, shown here in blue, 3 different lines, have fairly vigorous twitches. They get short and then they relax. And there's kind of puny twitches in these cardiomyopathic cell lines. So that was actually quite interesting. And and it turns out what happens is everybody has feeble contractions early on. And with time, the wild-type healthy cells get more mature, and they develop more vigorous contractions, and they leave the cardiomyopathic ones behind. And we were concerned, is this due to differences in calcium handling? We didn't imagine it would be. We thought it should be due to defects principally in the myofilaments. And in fact, calcium is the same. So it's a calcium-independent phenomenon. The next thing we did is made three-dimensional pieces of beating human heart muscle. We cast cardiac muscle cells into a gel of type 1 collagen doped in some basement membrane proteins and blah, blah, blah. You don't need to know all the details. I always describe this to my mother as our Jello mold experiment. I grew up eating a lot of Jello in North Dakota. And so the idea was basically the cardiomyocytes are going to play the role of the mandarin orange in the Jello. They're going to be suspended in this. And that surprisingly, you can make fairly respectable looking human heart tissue by just casting it into a gel. And it's hooked up to these elastomeric posts, and it's pulling on it. And heart muscle likes to work. It's one of the things that we found, that it makes just much healthier tissue if you let it work while it develops. And then you also need to throw in some connective tissue type cells, because there's kind of this mysterious crosstalk between fibroblasts and cardiomyocytes that makes tissue healthier as well. So we do that. We can make these beating pieces of human heart muscle in essentially unlimited quantities. And so Daniel took this and went down to Mike Renier's laboratory in bioengineering and wanted to see whether we could find differences between wild type and mutant heart tissue by doing sort of classical cardiac physiology experiments. And what we found is that we could again, this is pretty cool. We could do people remember Starling's law of the heart, which says that the greater the amount of stretch on a muscle fiber in diastole, the greater amount of force is produced during systole. So if I take in a deep breath, it draws more blood into my thorax. This increases my venous return, distends my ventricle, and so end-diastolic volume is up, end-diastolic stretch of the cells is up, and then you get a bigger beat for the next beat or two until homeostasis is restored. So this is a fundamental property of cardiac muscle, and it's one of the ways the heart regulates beat-to-beat contractile activity. What we found is that we could take these pieces of heart muscle and we could stretch them and measure the amount of force that's developed. And what we see is that we get a straight line curve, which is pretty cool, with the wild type cells, and the mutant cells are really pretty feeble here. They do show a positive Frank Starling relationship, but not nearly as vigorous as one would observe in the wild type cells. So an interesting thing here is we had to work really hard at the single cell level to see this defect. We put this into a piece of engineered heart muscle, and the first time we looked at it, we saw a threefold difference in the contractile power of the cells. And so this has us thinking that engineered heart muscle in the three-dimensional, much more native context is probably the way to go. I told this to Mike Renier. I said, Mike, we're delighted it came out like this. And he looks at me, he's just like, Chuck, you're just hooking the cells up in series. Of course you're going to see a much bigger difference than if you're just looking at a single cell. And I was like, wah, wah, wah. Okay, but no, we proved this, Mike. Yeah, that was just theoretical beforehand. And so apparently, things are never obvious to me, but that's why I'm an experimentalist. So, and here's an interesting thing. Let's go back. I told you about the way that there's this important protein-protein interaction that we're interested in chasing down. So if we look at a stereotypical twitch from one of our human heart muscle samples, so this would be actually, it's taking all comers, but so from the wild type shown at the top and from the mutant cells shown at the bottom, you can see again much greater force development in the wild type than there is in the mutant. People in Wisconsin had done similar kinds of experiments, not in the human, but in the mouse, looking at this myosin-binding protein C, where the mutation was in the opposite face of this protein-protein interaction. And they made engineered heart tissues and looked at their contractile function. This is what they found. So we're like, that's pretty much a phenocopy of using the human and the mouse, different proteins, but again, the same protein-protein interaction. You'll notice the time scale is different. Things are faster in the mouse because they've got a fast myosin, we've got a slow myosin, but everything else looks pretty similar, really. So that made us think that looking at this protein-protein interaction was a way to go to understand mechanism of what's going on with the disease. So we looked at, I'm going to dim the lights here for just a minute. Don't. Whoop, that's too dark. Sorry, there we go. This is looking at the architecture of the tissue using alpha-actinin as a Z-band marker. And we have reasonably aligned cardiomyocytes in 3 dimensions in the wild type, completely disheveled in the myosin mutants. And in fact, the histology reports on the sister who had the total artificial heart, when her heart came out, they described marked myofibrillar disarray. So maybe that's something we're reproducing in tissue engineering as well. That needs more exploration, I think. But we wanted to look at this protein-protein interaction. And so we worked with this group in Wisconsin. The guy's name is J. Carter Ralph. And basically what, sorry about this. Yeah, that'll work better. The way we're looking at protein-protein interactions through an assay called the yeast-2 hybrid assay. And essentially the way this works is you take a transcription factor and you break it up into two pieces. There's a DNA binding domain and what's called the transactivation domain. And the only way that you can make a functional transcription factor is to have your two proteins of interest interact inside the cell and physically bind to one another. It brings the two things together, and then the readout is it allows the yeast to grow in a nutrient-deficient medium, so that it changes their metabolism in such a way. So growth equals protein-protein interaction. So let me just jump to the most important stuff, which is shown here along in the bottom in the green box. So if we look for, what we're looking for is interactions between myosin in the S2 domain and the C1-C2 domain of myosin binding protein C. So when we use wild types of both, you can see we get nice yeast growth. When the C1-C2 domain is mutated, like the Ralph group had shown previously, it disrupts this protein-protein interaction and we get no yeast growth. What is interesting is here we've mutated the opposite face on our myosin, and this also disrupts the protein-protein interactions. So it's yeast, it's not human heart muscle, but this is pretty good evidence that this interface is readily disrupted by loss of just one of the salt bridges that forms between these two amino acids. So what have I told you in the first part of this? That we've been able to make human-induced pluripotent stem cells from a family that's got a myosin mutation, and it's a mutation that's associated with systolic dysfunction. We were able to differentiate these into cardiac muscle cells. We were able to align their myofibrils. And we found that there's essentially a phenocopy of the patients in the dish. These cells have impaired contractile kinetics, both at the level of single cells and at the level of engineered heart tissues. And our notion is that the Achilles heel here is this disruption of the myosin's ability to bind myosin-binding protein C. And what we're exploring now is, if that's true, if we could find a small molecule that would promote protein-protein interaction, we ought to be able to find a way to at least treat the cells and see if this restores contractile function. And ultimately, big dream, would be to use assays like this to understand on a personalized level how to treat patients as well. So we're just beginning to set up a mammalian 2 hybrid system that we can use in our high-throughput Quella screening facility to test for small molecules to promote protein-protein interactions. Okay, so that's the disease in a dish. And I'll just say by way of closing, I think this ability to study human cells of virtually any type now that we want with defined genetic backgrounds is going to have a major impact on genetics. I think this is going to be at least comparable to our ability to manipulate the genomes of mice. We can now, using technologies of genome editing, we don't even have to find the patients necessarily. We can just make it in the laboratory. We can have a standard generic, plain vanilla cell, and we can introduce whatever mutations we want using genomic editing technologies like CRISPR-Cas9, that sort of thing. And so it should be possible to make to understand, you get a hit in a GWAS study, for example, and you want to know what its physiological significance is on tubular epithelial transport, for example. It's a hypertension. It's a hypertension hit. It should be possible to engineer the mutation, derive the renal epithelial cells, and then see what effects it has on salt transport in a physiologically relevant cell type. So that's our idea. Okay, shift gears then and talk about cells as therapeutics. We've been at this game for a long time. And I'm going to save you all the pain and suffering. I'm going to summarize what we've learned in 15 years into one slide. And my students hate this when I do this, by the way, because they say, hey, that line is so-and-so's PhD thesis, but time marches on. So the first thing is that if you transplant cardiomyocytes into a myocardial infarction, they hate it. The myocardial infarction is an inhospitable environment for cardiomyocytes. They probably wouldn't have died in the first place if it were a hospitable environment. So we had to spend a long time learning how to keep our cells alive after we transplanted them. And we came up with what we call a pro-survival cocktail that blocks both apoptotic and necrotic pathways to keep the cells alive. We started, once we had the differentiation under control, we found out we could make grafts. We tested it in small animals. We began in mice. and we found if we put human cardiomyocytes into infarcted mouse hearts, that this would preserve the geometry of the heart, so it would prevent remodeling, and it would enhance the heart's contractile function. We moved up a factor of 10, went into the rat, found the same thing essentially, preserved geometry and enhanced contractile function. Then we moved up again about a factor of 4 or 5 into the guinea pig, and we found that the geometry of the heart was preserved, the contractile function was enhanced, and here we were able to start doing meaningful electrophysiological experiments. We actually found that the arrhythmias were prevented. And I should say, you start in the mouse, that's 600 beats a minute, the rat's 400 beats a minute, guinea pig's about 250. So 250 is just getting into the range of what we thought was physiologically relevant for human cells to really be able to beat on a one-to-one basis with them. And the final thing is that when the cells they have robust effects when we transplant them in the acute or the subacute setting. If we wait until we've got an old, scarred-up heart and the ventricles dilated and that sort of thing, we no longer have a therapeutic effect. We get engraftment, but we're not able to, so we can prevent heart failure from happening, but we cannot pull back the failing heart into a physiological functioning state. So this has implications in terms of how we want to transplant this. Though we'd love to be able to start in people who have old, burned-out heart failure and no other options, I think we probably won't be able to have a therapeutic benefit in such a patient, at least to the extent that these animal models predict. Okay, so that's the primer on the last 15 years. So what we decided to do was take this up, take our game up more than a notch, several notches, and see if we could actually do something in a model that I think would best reflect a human with heart disease. And so we decided to do this in non-human primates, and this is a magnificent creature that the most common species used in our primate center here called Macaca nemostrina, or the pigtail macaque. This is a 8 to 10 kilogram primate that's indigenous to Southeast Asia, and it's been used here for a lot of HIV studies, hematopoietic stem cell transplant studies, vision research, neurobiology. It's a tried and true model. There's a lot of expertise institutionally. So why do something as crazy as trying to regenerate a primate's heart? I would submit to you that this field is moving pretty slowly. We have had human embryonic stem cells since about 1998 was the first report. And I already told you the slow progression that we had made from mice up through guinea pigs. We were thinking maybe rabbit would be the next thing to test. And I was starting to feel like I was going to die of incrementalism before this thing ever got done. Essentially, I think we've been taking these little baby steps, and it was time to take a leap and see whether there was really promise to it. We've been working in the field of cell-based repair for a long time, and for reasons I won't get into. I really think cardiac muscle cells are the best singles. If you're going to talk of a single cell population to use, they're the cells that the heart is missing, and they're the cells that can provide new systolic force generating units. Most of the other cell types seem to work through murky paracrine mechanisms and that sort of thing. I don't think they're as robust. And I would submit that the non-human primate is probably the best model for human heart disease. It's certainly phylogenetically the closest. And I will make the following sort of arrogant statement. I think if we can regenerate the heart of a monkey, we can regenerate the heart of a human. So what I wanted to do was basically get to what folks in industry call a go, no-go point. If this was not going to work, I wanted to get that answer as quickly as possible so I could take my ladder and put it on a different wall and do something else for whatever, 15 years I've got left professionally, something like that. Conversely, if we could get a go signal, that would be meaningful and we could take our game up a notch. This is not something that a person does on their own. So for all of the rest of this presentation, if I say I mean we. If I say we, I mean they. You get the idea. I'm just the spokesperson here for this. And these are some of the key people. This is sort of like the section. On base, we've got, et cetera. But we've got James Chong, who is an MD PhD from Australia, actually a cardiologist. He's gone back to Australia now to finish his clinical training. He was the overall experimental lead. A long-term colleague, Mike Laflamme, led our efforts in cell production and electrophysiology. Mike's been a great partner in crime for me over the years. Creighton Don from Medicine Cardiology did our interventional cardiovascular work. And without Creighton, we'd have none of this. We had a big cell production team here. We see Ben Van Biber and Savannah Cook. Julian Yang was involved in our electrophysiological experiments, Veronica Maskelli doing our histology. And the Guy Fawkes mask is to show you the anonymous faces of our of our staff in the Primate Center. This is absolutely a terrific staff, but for reasons of security and so forth, I'm not allowed to show any pictures of the Primate Center staff. So I thank them anonymously for their efforts. There are a few in the audience here, but I'm not going to make eye contact to out anybody. Okay. So in order to do this, we did a variation on what our previous experiments had done. We made our cells, we affectionately call them beaters, but we made these beaters flash green every time they beat. And to show you this, I'm going to just drop the lights for a second. So what we did was to knock a calcium reporter gene into a safe harbor locus called AAVS1. And this calcium reporter gene, it's a variant of green fluorescent protein that's got a calmodulin domain on it that renders it calcium sensitive. And let's see if we can see it. Yeah, so you flip it to fluorescence, so you get this nice green flash every time the cells beat. And so this is like sort of cool screensaver stuff. But the idea was basically to do this in vivo and use this green flash as a index of as a reporter for what our human cardiomyocytes were doing. So we could answer the questions, are they beating? And if so, are they beating in synchrony or out of synchrony with the rest of the heart? So fairly straightforward question. Come on, there we go. Let's see how that works. Cell production. It took, here's back of the envelope math again. We'd done dose responses in mice, found it took about 1 million cells to repair the heart of a mouse. It took about 10 million cells to repair the heart of a rat. It took about 100 million cells to repair the heart of a guinea pig. Primate's about 10 times bigger than a guinea pig. So we sat around and we're talking about this, and I just said, guys, I think it's going to take us 1 billion cells to fix a monkey's heart. And they're like, my God, Chuck, no one's ever grown, no one's ever grown 1 billion cardiomyocytes. But we go around saying glibly that these are immortal, they're inexhaustible, they're self-renewing, blah, blah, blah. And so we should be able to scale it up. And so we went and did it. And we didn't do this through beautiful bioreactors. And there's all kinds of, there's a whole art of cell production that is sort of like chemical engineering. But we didn't do any of that art. We just did this with flat dishes and lots of surface area, basically. And so we had a very good-natured team involved in all of the cell production and just floor-to-ceiling incubators with the goal of deriving 1 billion cardiomyocytes per monkey that we're going to treat. And then we also had to develop a way to freeze these cells, because you can't have a really complicated cell production run going simultaneously with a really complicated animal model and just hope that you get them all to fruition at the same time. We proved that also. Okay, so here's the experiment. The animal comes in, and by the way, day 0 is considered the day of cell transplantation on this timeline. So about a month before the animals come in, they're trained to do what's called being on tether. This means that they wear a jacket and they are in their cage. And this jacket has it's a protection device that allows us to have chronic IV lines that exteriorize through the skin of their back, go up to connect to a swivel at the top of the cage, so the animal can move freely about the cage. It's one of the real advantages of working in the Primate Center, because this has again been worked out for all the hematopoiesis work that Hans-Peter Kiem's group has done and so forth. So we bring them in, teach them to wear the jacket and behave well on tether. We had a council of wise people, and the council said better start animals on antiarrhythmics just in case, so we put the animals on amiodarone. And then at day -14, we do the infarct. I'll show you how we did that in a minute. We also instrument the animals for chronic telemetry so that we can follow their electrocardiogram 24/7. These are human cells going into a monkey, and we knew that these would be rejected if we didn't immune suppress. So we did triple drug immunosuppression, really heavy immunosuppression. If people are interested, we can talk about that. And again, fortunately, we were able to take a whole cassette of knowledge from the groups who have been doing hematopoietic cell transplantation. So we knew how to keep these animals alive without infectious complications and things like that in the Primate Center. So then we transplant them at day 0, and we studied the animals. We monitored them along the way, and we studied them at 2 weeks, 4 weeks, and 3 months. And the endpoints are sort of general animal well-being. They're monitored regularly by staff. There's also 24/7 video surveillance. We were able to, as I mentioned, track the animal's heart rhythm on a 24/7 basis using telemetry. And then after the sacrifice, we're going to take the hearts out and study those physiologically for evidence of electrical integration of the grafts. We'll do angiograms and histology. And I'll show you just the highlights of that, because there's not time in today's lecture to get into all of this. So the first thing we do is to take the primate center's very nice surgical suite and turn it into an interventional cardiology laboratory. First we turned it into a coronary catheterization laboratory. Later we turned it into an electrophysiology laboratory. People will recognize this isn't quite human standards, but it's pretty good. This is the silhouette from a fluoro image of the monkey's heart. And a monkey's heart's about the size of a plum. So it's not, we're not talking a human size yet, but it's a good-sized heart. We've got, Creighton's put a catheter up the aorta. Here it's cannulating the coronary ostia. We've got a wire down the left circumflex for anatomical reference, and then we've got a wire down the LAD. And this is a balloon inflated in the distal LAD, and that's how we create our infarct. So it's a 90-minute inflation of the balloon, creates an infarct, marches about halfway through the wall, and then we drop the balloon and reperfuse. And this is a shot of what the precordial electrocardiogram looks like. You can see a very nice ST segment elevation, like a patient coming in through the emergency department doors with a STEMI. So I think this is a very good model of human myocardial infarction. 2 weeks later, we do a second procedure, and this is the cell delivery procedure. In patients, we're going to use catheter-based delivery technologies. We didn't have that working in the monkey yet. I didn't want to, for this go, no-go experiment, I wanted to do this by directly visualized cell injection with a simple needle and syringe. So we performed a thoracotomy. This is the lead veterinary surgeon, and this is yours truly holding a very expensive syringe full of human stem cell-derived cardiomyocytes. And we transplanted these by, again, with just a needle and syringe into the infarct region and peri-infarct region. So here we're looking down into the surgical wound. We see this white thing that's going along in the left margin. That's the left anterior descending coronary. The LAD was occluded probably right about here, so this would be the zone of infarction. And we transplanted the cells into the central ischemic region and in the peri-infarct region. And again, about 1 billion of them in a volume of about 1.5 milliliters. Okay. So the animals are monitored, and then they're euthanized at the appointed time. And we take a page out of the cardiac surgery books where we perfuse a cardioplegia solution. And so we arrest the hearts indiastole with a high-potassium cardioplegia solution, put the hearts on ice, transfer them from the primate center down to our laboratory at South Lake Union to do the calcium imaging experiments. We're going to do this in what's called Langendorf mode. So people may be familiar that the heart will beat outside the body for hours and hours happily if you provide it with the crystalloid buffer that's got the right salt concentrations and some glucose for it to eat. And so people have done this a lot in mouse and rat. I haven't seen anybody do this in a monkey, but we figured, why not? It should scale. And so this is Julian just showing you the liters and liters of perfusion buffer that we had to do to make this thing work. This is kind of a fun, a slightly macabre movie to start the morning, but it is just after Halloween. But we're looking at the monkey heart that's being perfused, and we'll start this just as we're switching out from the cardioplegia buffer to a normal thyroid solution that has normal sodium, potassium, and calcium balance to it. What you're seeing is a couple of recording and pacing electrodes, one in the atrium, one in the ventricle. And then this is the hose that goes into the aorta to perfuse the coronary circulation that will be delivering the buffer. And you'll just notice that this is the little white thing here is a patch of pericardium. There's a little pericardial adhesion from the surgery. So we can pan back, and this is the hose going into the aorta. This is a heat exchanger to make it at 37 degrees centigrade. And then it's a simple system, just two large gravity-fed reservoirs that you'll see, essentially carboys sitting up here. Over to the right are our fancy schmancy, highly expensive and sensitive cameras to do the calcium imaging. Here's the cheap tape to tell you what's red and what's green. And then that's all hooked up onto this fluorescent stereo dissecting microscope that we use for the imaging apparatus. So let me get back to... So while I've been blathering, the heart's been getting itself going, so it's got a pretty good facsimile of sinus rhythm now. We let it stabilize for about 15 minutes, and then we stop the mechanical activity because when the heart's jumping around, it's really hard to do calcium imaging because things come in and out of plane. So we stop the mechanical activity with a drug called BDM or blebistatin, and then but the electrical activity persists. So that's what we're going to look at here. Again, I'm going to dim the lights just 1 second. Apologize for doing this, but it's easier to see what I'm going to show you. We're looking at the apex of the heart, and these white streaky things are areas of human grafts where their fluorescence is shining through the epicardial surface. If I turn the movie on, if you look at this one up here, it takes your eyes a minute to get used to this, but you can actually see there's cyclic fluorescent signal that's coming out of it. And once you see that, you can start seeing it over here, and over here, and down here, and down here. And really, there's a lot of action going on in the apex. So it looks like there's a lot of human cardiac muscle. And in the first approximation, if the question is, does this stuff beat after you transplant it, I think the answer is yes, it does, because we can see this fluorescent signal. The next question really is whether it beats in or out of sync with the monkey's heart. And so we draw, the way we do this analysis is to, sorry, I'm having a hard time getting my cursor to come up here, is we get we draw regions of interest on the video tracing, and we look at the light intensity signal. And so what you can see, these are the calcium waves from 1, 2, 3 different regions, and this just tells you what your eye told you already. They're pretty much flashing in sync with one another. The important thing, this is the electrocardiogram along the bottom, and the signal here is generated by the monkey's myocardium, and you can see every QRS of the EKG triggers one of these human calcium transients. So this is physiologically very strong evidence that our human cardiomyocytes have electrically integrated and are beating in synchrony with the monkey's heart, which is really a sine qua non for regeneration. We can pace from the atrium, and the human graphs will follow. So you're looking at the calcium transients of the human on the top, the electrocardiogram from the monkey heart. This is spontaneous. Note when we turn on the pacer, you get a different QRS morphology, but the monkey follows up to at least 180. We're getting kind of this funky alternans stuff when we get it up to 240. But I figure, 180 is pretty good. That's about as fast as I get my heart rate up to these days anyway. So that's not too bad. What do things look like structurally? If we, this is a confocal micrograph of the monkey's left ventricle. The monkey myocardium is shown with a cardiac muscle marker called alpha-actinin in red. The region I've masked out in blue is the zone of infarction. And if I take away, so it's a decent-sized infarct, at least towards the apex. If I take away the mask, this is the region that we've been able to remuscularize with our human cardiac muscle graft. So you can see it occupies 40 or 50% in this particular image of the infarcted region. Here's some that leaked out and grew a little nodule of human cardiac muscle on the surface of the heart. And remember, I showed you that piece of adherent pericardium. This is it right here in histology. So there's like a pericardial adhesion over the top of that little graft spot. This wasn't just one lucky shot. We were able to see this in multiple regions. Here's from the peri-infarct region. So this monkey had a lot of human myocardium within it. And if we did the mathematics, we were able to remuscularize about 40% of the infarct mass in a series of 4 monkeys. And I would submit if we could do this in a patient, if we could remuscularize 40% of their infarct, that would probably have meaningful physiological impact on their left ventricular function and maybe downstream for clinically harder endpoints as well. One of the criticisms of these has been that the cells are immature and that they may not mature. That's a bunch of hooey, I can just tell you at the bottom. They turn into nice hypertrophic-looking human cardiomyocytes, and this is just the data, quantitative data to show it. For reasons of time, I won't belabor it, but they mature nicely inside you. We have done angiograms of these hearts postmortem. So this is a CT angiogram, and the LAD bed is shown in red, and then the area that we've shown histologically is involved in the human myocardial graft at the apex is shown down at the bottom. So there at least is arterial input into the grafts. It doesn't prove that it doesn't prove that we've got great flow. It just says that structurally there are arteries within the region. We don't know if they were there before or not, that sort of thing. It's just a snapshot, but at least it's evidence that we've got some blood vessels in the vicinity of the graft. I showed you that they were beating in synchrony, so there must be a basis for electrical coupling, and the way that the heart is coupled normally is with gap junctions of connexin 43, and so here's the interface between graft and host with connexin 43 positive gap junctions. All right, so here's the dark side, and this is where I'll wind up. Every animal we put the human cardios into had ventricular arrhythmias. So you can see this is stereotypically what happens. They're in sinus rhythm. Monkey's heart is about 120 beats a minute. We start to get these runs of non-sustained VTach, periods of sustained VTach, and then they go into accelerated idioventricular rhythm, and then typically they come out of it. But it's serious. We look at the VTach. So a couple of animals had sustained VTach. A bunch of animals had non-sustained VTach. And it looks like by 3 weeks or so, they're coming out of the woods. This is the accelerated idioventricular rhythm. This animal took about 4 weeks to come out of the woods. But we took one animal out to 3 months, and for the last 2 months, it had not a blip on its electrocardiogram. So what we think, period of electrical instability as the heart is rewiring and remuscularizing, this heals in, the cells adapt, and the arrhythmias go away. Clearly undesirable for our patients, however. So we needed to map this. And I was talking with Rob McClellan, and I was like, Rob, where can we find somebody who can do endocardial mapping of a 10-kilogram primate heart, the heart of a 10-kilogram primate? And Rob says, Chuck, I know people who map hearts of 10-kilogram primates all the time. They work at Children's. and indeed they do. So we found this guy named Steve Sessler, who people may know. And this is work that's been also led on our side by Yenwen Liu, who's a very talented postdoc in the laboratory. And so Steve and Yenwen worked together and figured out how to do electrical mapping of these monkey hearts. And let me just show you what this looks like. This is a happy, healthy primate heart. And you'll see the activation. It comes from the apex of the heart and spreads up towards the base, just like a human heart does. So that's normal, happy cardiac activation. Here's what happens in one of the animals that came in ventricular tachycardia. The apex is down here, and the hot spot where it activates is actually in the anterior septum in probably a region where we have our grafts. I can't prove that 100%, but that's what I think. And if you look at the activation, it spreads out from up here and goes around the rest of the heart, goes down to the apex, and so it's like activating halfway up. And based on the putting it through the paces, literally, Steve's impression is that this isn't reentry, which is what all my money was on. It looks like it's acting like an ectopic pacemaker. Interesting. So that's just a comparison of the activation maps. I think for reasons of time, I'll just wind up here instead. So this is where things stand at the moment. It's a work in progress. We're not quite there yet. I think we're close, but we've got to work on this electrical issue. So we've got this non-human primate model for heart regeneration. We've done this with a couple of stem cell lines. These have been the largest grafts of human cardiac muscle anybody's been able to make to date. It's tenfold better than the previous record. I know that because we had the previous record. It's coupled extensively electromechanically between the graft and the host. But that, in turn, is a double-edged sword, because I think that's this electrical coupling and the rewiring of the heart is also the basis of electrical instability. These have been non-fatal to date, and they have been transient, but these are previously healthy animals, not diabetic, elderly, frail, etc. So I don't take this at all lightly. Our preliminary studies suggest that this problem is ectopy. So clearly, we're going to need to figure out what to do to manage these arrhythmias en route to any clinical translation. And the last slide I'll show you is just to tell you that the school actually has bought in on this. So I thank Uncle Paul Ramsey, for recognizing that this actually has some promise for human heart therapy. So we've created this entity called the Heart Regeneration Program, the objective of which is to do a first-in-human clinical trial of stem cell-based cardiac repair, and we'd like to do that here in Seattle. So Rob McClellan, Mike LaFlamme, Scott Thies, and I are the main directors of this program. And it involves multiple partners across the university, usual kinds of thing. Again, it takes a big team to make this stuff work. And the evil plan, if you will, is to create like a little bubble, almost like a biotechnology company within the University of Washington. So we've been hiring people from industry to work on this. And from a clinical standpoint, our target population is folks with ST segment elevation myocardial infarctions, people who have large infarcts and low ejection fractions. And the timeline that we see for this is about 4 years if we push aggressively, which is what we plan to do. So that's it. I'd just like to say thanks to the people in my laboratory who made this possible. I get individual shout-outs along the way, so I won't call people out individually. I'm just really proud to represent the work of these people. It's an absolutely terrific bunch, and I'm fortunate to have them affiliated with me. And the folks also, it's a very collaborative environment at the University of Washington, folks within our Center for Cardiovascular Biology, within our Stem Cell Institute, very valuable folks in our Primate Center without whom we couldn't get things done, folks at the Hutch and so forth. We also thank the American taxpayers for supporting this work. And if there's any time left, I'd be happy to take a few questions. Thanks a lot for your attention.
Absolutely terrific. Thanks. Not a hard question, but did any of the monkeys die, either from surgical complications or from, later post-op?
So the question was, did any of the animals die? We've had one animal die from an iatrogenic complication where we came in and tried to suppress the arrhythmia with verapamil. And this turned out to have been a bad idea because the animal was in VTAC. We gave it verapamil, and I think it massively vasodilated. And my cardiology colleagues were like, "You gave what?" And so that was the one death that we have had that was unplanned, that was related to the problem. So it didn't die spontaneously. It died because somebody had a brilliant idea. Yeah.
So these graphs are by definition going into ischemic areas, and you showed some pictures that suggest maybe microvasculature regrowing into there. Are you going to need some more macrovascular approach to get blood to these graphs?
That is a great question. And the answer is, heck yeah, we do need that. Our best evidence suggests that if you look under the microscope like a pathologist does, you see a fair vascular density and erythrocytes in the vessels, and you think, oh, life's good. But if you think like a physiologist and measure the dynamics of flow, what we found is that it's sluggish. So I think it's mostly angiogenesis from one postcapillary venule to another postcapillary venule, and it's kind of like wetlands. And so what we need is arteries. And so we're working with vascular biologists on strategies to grow in new arteries. That's been a challenge for a long time, of course. So we're not predicating our going forward based on that, but that's a big challenge. Thank you for bringing it up.
Excellent talk. Before you go into human trials, do you have ideas about approaching the issue of rejection and reducing the need for anti-rejection medications?
Right. So that's of course the immunosuppression is the bane of transplantation medicine, and yet we're willing to live with it, I guess, because we transplant every heart that we can. I'd much rather not transplant. I would much rather not immunosuppress our patients. And so, just down to your left is David Russell, who is developing new technologies for making what we're calling universal donor cells, or what David's calling that. And the idea there is to knock out class 1 and class 2 major histocompatibility complexes and to make essentially the equivalent of type O blood that could go into anybody, or at least with markedly less immunosuppression. I'll give David a shout out. He's getting the university's Inventor of the Year award today at South Lake Union in recognition of the groundbreaking work he's done on that. So we're looking to partner with David. So it's like, David, fix the immune system. We've got the heart. You deal with immunity.
Chuck, over here.
Oh, hello, David.
Hey, so very nice talk. With replacing 40% of the infarct, do you expect an effect on global LV function? And did you?
So I would expect an effect on global LV function. It's essentially like a 40% reduction in infarct size, and we certainly know based on reperfusion studies, if you reduce infarct size that much, you have a marked effect on overall left ventricular function. We looked for it, but we did a poor job of it in retrospect. We tried this by transesophageal echo, and the primate heart sits differently in the chest compared to the human, blah, blah, blah. I won't get to, but we're like standing on our heads trying to shine the sound beam through this transgastric thing across the liver to look at the heart. We got lousy functional data on that. So what we're doing now is MRIs, and that's a gold standard level measurement. So just one of the criticisms I'll throw at my own work is that we didn't do a good job at assessing systolic function in these animals, and we're doing it now, but it's a work in progress.
One of the early challenges with embryonic stem cells was teratoma formation when in vitro plants. Is that a problem with your induced pluripotent stem cells?
So we remain vigilant because at the moment it looks like we've basically overcome the problem with teratomas. Teratomas is one gets a teratoma when you don't have good control of differentiation and you transplant in still undifferentiated pluripotent stem cells. If you have good control of differentiation, you should not get teratomas. The rub is as you scale up the number of cells you put into billions, then could a few little lurking pluripotent guys do it? And so that's why we've done thousands of rodents and we don't see teratomas. We've done many hundreds of guinea pigs and we don't see teratomas. Occasionally we'll see a funky epithelial cyst. That's about the worst of it. Not that that's trivial. We would like to get rid of that too. But the worry about the frank teratoma with teeth and hair and all that kind of stuff has just not come to pass.
Thank you.
Good. Thanks very much.